We'll wait for a couple more minutes, then start. Okay, so let's get started. So, welcome to week 3 of bioinformatics algorithms and applications. So, the first class was more of an introductory class, then we.
Sir, you are on mute.
Oh, okay, sorry. Okay, so welcome to week 3 of bioinformatics algorithms and applications. So, in the first class was more of an introductory class. In the last class, we talked about uh, different types of methods by which you can do sequence alignment, mostly dot plots and other scoring matrices that you can use. Today, we are going to build on that and talk a little bit more in details about these types of alignments and yeah, an example of with respect to an example of blast. So okay, let's get started. I think somebody's waiting. Okay. Okay. So yeah, why do we need scoring matrices? We talked about this in the last class. So every substitution, there are 20 amino acids, and all the possible combinations that can happen while substituting one amino acid inside a long chain of polypeptide protein. Is like a lot of combinations can happen. So each of the substitution hold a different impact on the protein itself. So if you are replacing a particular amino acid with another amino acid of similar nature, probably it does not affect much of the protein in terms of its structure and folding, or with respect to its active site even. But if you are if a particular amino acid is replaced by another amino acid which is different in nature, so for example you are replacing a positively charged amino acid with a negatively charged amino acid or an aromatic amino acid, then the impact is much more. So our scoring matrices need to reflect that. So as the name of the slide says, the cost of the mutation is not constant, right? So same for DNA, where we have different types of substitutions, transitions and transversions, which are like purine to pyrimidine or purine to pyrimidine, and the transversion is purine to pyrimidine or vice versa, right? So different type of weights are given to these different changes, depending on what type of change it is, right? And same is for proteins and amino acid. I was saying, say for example, you are converting by mutation, a lysine has been converted to an arginine, but it won't affect much because both are similar in terms of their chemical nature. I swear. Okay, so yeah, lysine and arginine are very similar in nature. Both are positively charged. Isoleucine and leucine are also similar in nature in the structures. So are serine, threonine, or aspartame, glutamic acid, or the other amino acids, right? Okay. Um, but the general idea is that if your side chain chemistry is similar, then the mutation is less likely to change structure or function. So if you're by mutation, a positively charged amino acid is replaced by a positively charged amino acid, the chances that it will change it, it will change the structure or function of the protein is very low, but it's not completely zero. It is possible that you can have similar amino acids mutated, but the structure or the function change, specifically for very sensitive enzyme, very sensitive proteins like enzymes, where the active site is very sensitive to the amino acid residues present in the active site, so that they can bind to the substrate, right? So even if you changed a particular amino acid which is very similar in nature to the one that was there, it is possible in some cases where the specificity is very high that the enzyme cannot bind to the substrate. So that can also happen. It's not like a binary thing, but mostly it won't, like in most cases. So here is one example of such a conserved protein. So histone, as I think I was talking about histone in last class as well. So histones are positively charged proteins which are multimeric in nature. It has 8 subunits, makes it an octamer protein, and it is positively charged in nature due to the fact that the DNA has to wrap around that histone to form different chromatin regions in the cell, in the nucleus. So where that is most densely packed, it is the euchromatin, and when there is not much packing, it is called heterochromatin. So different regions of the DNA is packed differently, and that is done wholly through these histone proteins. Now these histone proteins are very conserved in across evolution. The sequence similarity of histone proteins of humans and their ancestors, like if you look it for in a chimp or a chimpanzee, or different types of apes, or even other families of animals like rats and cows and mice, the amino acid residues are very similar in nature, and it is very conserved across evolution. And there are some sites which are much more conserved than other. So for example, if you check this set of amino acid residues, they are more conserved in nature, and if you check the ones that are highlighted, those are less conserved. But these set of amino acids that are less conserved provide specificity to each of these organisms' histone proteins. So some of these variable regions are conservative in nature. So if you change those amino acids in those regions, are most probably your histone is going to function as slightly differently, which happens in different organisms. And there are some regions where if you put in like a similar type of a mutation, it won't change much of the structure or the function of the protein, so it is non-conservative in nature. So there are different substitutions that can lead to this conservative or non-conservative nature of histones. So these are the set of combinations that can happen. So if you have a valine residue, the conservative substitution for the valine residue would be alanine or phenylalanine. Or if you want to do a non-conservative substitution, those would include lysine, aspartate, and threonine, right? So different type of combinations can happen, and these different combinations uh, can have different impacts on the protein structure of the protein or the function of the protein itself. So this protein histone is very well studied across most organisms, and this kind of analysis has been done on data of histone, and we have found different combinations which are conservative in nature and non-conservative in nature. This is to give you an example about how like what kind of variations that can happen, uh, keeping the function
Sir,
of.
how you understand what is how you are distinguishing between non-conservative and semi-conservative, sir?
So say
They
here,
are looking same.
it the non-conservative and the semi-conservative is same, except the last residue, I think, if I'm not wrong. The T and A are different. So the thing is, just a second. Yeah, so semi-conservative would ideally be something that is not fully changing the structure of the function, but it is affecting to a certain extent to which you can see a difference with respect to the wild type, right? So those would be categorized as semi-conservative. But for simplicity, you can imagine a particular site of amino acid residues to be conserved or non-conserved. So semi-conservative would sit somewhere in between, where some of the functions are altered, some of them are not, probably, or the structure is slightly altered, and in some cases it is not. So I wouldn't weigh too much on the non-conservative part. I can give you the paper. I'll probably link this paper in the chat box later. There's a very good study done on this set of histones, and what amino acids are conserved across organisms and what are not. And these are mostly computational predictions. So you'll have a computational model where basically a change in the amino acid, as we do in sequence alignment, right? So if there is a change, can you predict if the change will do something to the protein structure or function or not, depending on different scoring matrices. We use PAM or Blossom or things like this. But in this case, it is a little bit more complicated than they have checked. They've made a model out of which they have put in these different combinations and tried to check what are the combinations that can preserve the function, what are the combinations that can like fully alter the function, and what are the combinations that can kind of preserve some functions and alter the others. So things like this. So yeah, the to answer your question, what is the difference is basically, I think, the last residue is different in terms of the semi-conservative nature.
Yes. Thank you, sir.
Okay. Okay, moving on. As I was saying, for nucleic acid and proteins, for both DNA and protein, we have different alignment scoring matrices, and which is basically a set of values representing the likelihood of one residue being substituted by another during sequence divergence through evolution. And we call these a substitution matrix, and PAM and Blossom are kind of examples of these. Now, coming to the concept of sequence alignment, we have talked about sequence alignment in a very general manner across last class and a little bit of today as well. But formally, we define sequence alignment is by the fact that you have multiple sequences of amino acids that make up a protein, or multiple sequences of DNA that make up a region or a gene in the genome. And what you try to do is arrange these two or more set of these sequences to identify regions of similarity or differences between these sequences. The, uh, the regions of similarity are helpful in understanding the functional, structural, and evolutionary relationship between these sequences. Now, uh, I just want to make you think intuitively in terms of, if you imagine, say, for example, in uh, this set of amino acid residues are fully conserved across various organisms of different ancestry, right? So human and chimp are close ancestors, but human and cow are not very close ancestors. But this chunk of these residues that make up this part of the protein is very well conserved across different organisms. Could you imagine why? The why would, if you try to imagine why, the answer would be that histone is a very fundamental protein in any organism, in any eukaryote. Particularly prokaryotes, we do not have those nuclear packaging and those kind of things, but in eukaryotes, histones are very important, and it's fundamental to how genetic information is replicated, it's translated. The whole central dogma of biology depends uh, on the nature of these histone proteins, and if the DNA is accessible due to these histone proteins in those particular regions of the DNA, right? So histone is a very fundamental protein that is present in different eukaryotic organisms, and the function is more or less similar across different organisms, right? So the function basically is to wrap the DNA around it, right? So it's a very simple function, but it is well conserved across different organisms. So the idea is that if you have a chunk of these residues that are very conserved across a lot of organisms, you would try to you would make an inference that this particular residue block is probably very important to the structure or the function of the protein itself, because that structure and function is more or less conserved across different organisms, and correlating to that, a chunk of these residues are also conserved. So the first hypothesis that would come to mind would be that this probably helps these residues are probably in the active sites, or like these are residues that are outside of the histone protein that actually provide the positive charge, so that the DNA can bind to the histone, and so on and so forth. I'm just about the functions, I'm just giving you examples, but generally this would be a way to understand why these regions have been conserved. So the idea is that the similarity or differences of different sequences across, here we are comparing the same protein, in some cases we can compare different proteins as well, so they can tell you a lot about the structural and the functional relationship of those residues with the protein itself, and what are the kind of evolutionary relationships that particular set of residues have across different generations of different organisms, right? So the sites that are getting mutated easily, they are less conserved, so there is a less chance that they are directly involved in the primary function of the protein or primary structure of the protein. So those residues can be mutated, and it won't change much of the protein itself. So that is the idea, right? So sequence alignment is a very important aspect in bioinformatics across different domains, right? Be it proteins, be it nucleic acids, be it mRNA, be it whatever, like sequence alignment is central to a lot of things that you do in bioinformatics, right? So two types of alignment we'll be talking about in today's class as well. So those are like the global alignment and the local alignment. So what is global alignment? It is basically you're comparing two sequence, but what you are trying to do is basically you're trying to compare the whole of one sequence to the whole of the other. So, and what in the meantime what you're trying to do is you're trying to maximize the overall similarity. uh, if you have sequences of same length, so for example, as I was showing in the example, if you have sequences of histone protein across different organisms, most of like almost all of them will have the same length. So in that case, you can do global alignment. Basically, you are trying to match the whole sequence with another sequence, right? But in case of local alignment, instead of trying to align the entire length of sequence to another sequence, what you do is uh, you try to match only the regions with the highest density. Like so what you're trying to do is, if you have two sequences, one is of, say for example, 1000 in length, and another is of 400 in length, right? By like common sense, the 400 in length is probably not the whole, representing the whole 1000 base pair length sequence, right? So the idea is that you do not want to align the 400 base pairs all across the 1000 base pairs for your other sequence, right? What you want to do is you want to find a region in the 1000 base pair where this 400 base pairs correctly match, and not for the whole sequence. So this is very useful in identifying short regions of conserved regions in proteins and different nucleotide sequences as well. So if there is a very high length difference in the sequences that you're trying to align, local alignment is kind of the go-to tool, and mostly what we do in daily life is local alignment, because global alignment is not something that comes up very often, right? If you're comparing similar sequences across different organisms, like the histone example that I showed, there probably global alignment is the way to go. But in most cases, what you'll be doing is aligning a shorter sequence to a longer one. So that in that case, local alignment is the uh, is the accepted method, right? So this is just to show like pictorial representation in terms of the difference with respect to global and local alignment. Um, yeah, as you can see, this is the whole string length, right? String. If you have two strings like this, and you're trying to, in global alignment, you're trying to match the entirety of the first string with the entirety of the second string, right? So in that case, you can see this is the alignment that is the most optimal, and here you can see there are a lot of gaps yeah, here on the left, on the right, and you can understand that probably these two sequences do not have a same origin, or these two sequences are not very similar in nature. But there's a small region inside the first and the second sequence which are very similar in nature, which is this part, these three base pairs which are like one after another, and they are very similar in nature. And so if you just want to see what is the maximum similarity of a short sequence with respect to the longer one, so in that case local alignment works better. There's another example, I think, in later in my slide, where I've shown that if you use global alignment while doing an alignment of a sequence which is very long and a short sequence, global alignment actually fares very bad when you compare it to local alignment. So we'll talk about these examples when we go on. Right. So in terms of types of sequence alignment, we have majorly two categories in terms of how many alignments we are doing. So first one would be pairwise alignment, which is simple in nature. You have a pair of sequences and you are trying to see how similar these pairs are, and you want to find the optimal pairing of these sequences. And it is based on a scoring system that assigns positive scores to matching characters and negative scores to mismatching characters or gaps, as we discussed last time about affine gap penalty and those kind of things. So all those things are very relevant here. So and the main objective of a pairwise alignment is to obtain the highest possible score. If you have two sequences, what you're trying to do is maximize the match between sequence 1 and sequence 2 with different alignments, right? So that is your objective. Your objective is to have the highest possible score, which would be proportional to the degree of similarity between these two sequences, right? Which is like all the examples that we have discussed mostly are pairwise alignments. But in some cases, what you would want to do is try to compare multiple sequences, more than three or more sequences together. As I was showing in the histone example, you are comparing sequences of 4 different organisms together, right? uh, so here the idea is to find conserved sequence regions and to construct phylogenetic trees. I don't think we have covered phylogenetic trees here, but the general idea of phylogenetic tree is basically, if you have a tree-like structure, the the branch that forms the two, say for example, do I have any? Just let me go through it once. If I have any phylogenetic tree example, I think we have it here. So this is what a phylogenetic tree looks like, right? So what it means is, like, A is most similar to B, and these two are most similar to C and D. Now C and D are more similar to, say, C is more similar to D compared to B, and D is the least similar across all five. So this is what a phylogenetic tree would look like in terms of the relationship between any two pair of sequences, right? Now let's go back here. Yeah. So the idea would be to find, like, the objective would be to find conserved sequence regions and construct phylogenetic trees based on how much similarity is there between any two sequences in the whole set. And as I was discussing, it would help us to understand the functional and the evolutionary relationship between different species or group of organisms, right? Now, in terms of pairwise alignment, we've discussed this dot matrix method. It's basically you have you write the sequences as the rows and the columns of a matrix, and you have dots where there are matches, and blanks where there are no matches in itself. And ideally, if two sequences are very similar, you should have a diagonal line representing that. In this case, this is also very similar. You have five matches here, another I think six matches here, no, six matches here and six matches here. But if you have parallel lines to this diagonal, what it means is there are a lot of repetitive regions. So what it means is, say for example, this is how, what is the position, 1 2 3 4 5 6 7 8. So 8 9 10 11, yeah, 8 9 10 11, these 4 points are very similar to these four, which is 1 2 3, sorry, 1 2 3 4, this is fourth, fifth. Yeah, so you see this region. So okay, just a second. Yeah, so you have TCTA here, and you have TCTA here. So this is matching, this match is given by this particular line, and this match is also given by this particular line, which are parallel in nature. So if you have parallel lines to the diagonal in a dot matrix plot, it indicates that there is some repetitive region in and around that particular parallel lines, right? So yeah, we have discussed dot matrix dot plots in the earlier class. I won't go into much of the details, but yeah, just a recap that if it is very similar, the sequences, you will have a clean diagonal line. If yeah, if it is different, you would have a scattered like kind of a plot. In this case, mostly it is. If you have parallel lines to the diagonal, that indicates there are repetitive regions. And if you have opposite diagonal lines, meaning all the diagonals are in the leading diagonal, if you have lines in the opposite diagonal, what it means is there are inverted sequences as well. So just to keep a few things in mind. Okay, now how do we actually do the alignment? How do we know that pairwise alignment is two sequences with uh, for which we are trying to find the maximum similarity score and so on, but how do we do it? So the most widely used method is a dynamic programming method. So it's, this name sounds uh, very scary, but actually the idea is that if you are comparing two very big sequences, the idea is that you do not do it all at once. Like you do not start from position 1 in sequence 1 and position 1 in sequence 2, and then you continue to do the alignment to the right. It does not happen like that, because it's a very big sequence and you need to try a lot of combinations to find the perfect alignment score. So what they do is they try to break it down into smaller problems. So they try to break it down into smaller regions, uh, for which you can do the alignment easily, and then add all these information to find the best possible alignment, right? So the idea is to keep a reasonable bound on how many of these sub problems we have to solve, because you're breaking down the bigger problem into smaller problems. And also we need to make sure that each sub problem is solved only once. There is no repeat, in terms of like when you are doing an alignment of say base pair A to base pair B, uh, that region should be aligned only once. Now you do not have to take some middle point in these two base pair regions and then again do an alignment. That is not required. You choose a block and only the alignment has to be done only once, right? I'll just quickly go over the algorithm as well. So if you have two sequences like this, S and Sj, so A1 A2 An and A1 A2 till Aj, the idea is that uh, for you need to align till the ith position in S is in Sj, and in till the jth position in the second sequence. So S and S prime are the two sequences. So ith position of the first sequence to the jth position of the second sequence. So the idea is, the problem is to find the score of the highest alignment between these two regions of these two sequences, right? Now, how do you do that? You take the help of a matrix, scoring matrix, where which will give you the similarity score between any two pair of amino acids, which would be, for example, PAM or Blossom. And you also would need a gap penalty to to tell, to penalize for introducing gaps in the alignment, right? Excuse me. Now, this entity, so the idea is that you, I think, yeah, I think I can explain it here. So this is the ith position on the left, of the left sequence, and this is the jth position of the top sequence, right? So by definition, what is the alignment score of this position, I comma j? That would be, if the maximum of these three. So what are the three? So you align s I minus 1 and j minus 1. So there will be some score here, because if, while reaching to the ith and the jth position of the first and the second sequences respectively, you would have computed the j minus 1 and the I minus 1 score as well. So for any I and j in the matrix, you compute the I minus 1 and the j minus 1 score here, and then you add the score for what is the substitution here. So from here you are trying to move here, right? So the red one is representing the first score, right? So you have the alignment score over here, and then you add another substitution score for that particular base change, right? So that would be the first option. The second option would be to assign a gap from the left or from the right. So if you want to assign a gap from the left, so it would be I minus 1. So this is the ith position, I and j. So I minus 1 would be this position, the blue one on the top. And the idea would be that your I, so Si and Sj has matched till I minus 1 comma Sj, and there is a gap at this position. So you compute this alignment score till here, and you introduce a gap to reach here. Similarly, from the left hand side, you align till I and j minus 1, and then you add a gap to be present here. So these are the three possible things that can be present. If you imagine, while doing a pairwise alignment between any two base pairs, what can be the possible aligning, right? First, that there is a match. Second, that there is a mismatch. The match and the mismatch scores are taken care of by the scoring matrices. For matching score, you'll have some fixed score, and for mismatch, depending on the scoring matrix, you'll have a score for the mismatch. And the third thing that can happen is there can be a gap. Now the gap can be present in the top sequence, the sequence that is given by the column headers, or the side sequence which is given by the row names, right? So gap can be present in either. So you compute the possibility that there can be gap in both the sequences, and you compute their values, and you also assume that there can be a mismatch or a match, whatever is that in that particular region, and you compute all these three scores. The idea is to have the maximum score to be put in this particular box I comma j, which will tell you that if this is the maximum, then you put the score of this here. If this is the maximum, you put the score of this here, and so on and so forth, right? Okay, I thought somebody was, yeah. Okay, now the idea is simple in terms of that these are the possibilities that can happen. There can be a match, which is very easy to handle. There can be a mismatch. We'll handle it using different scoring methods, scoring matrices. And there can be a gap in either of I or in J. Like both of them can happen. And when you compute the scores for each of these three cases, what you choose is basically the maximum out of these three. The maximum score will tell you basically which one of them is more plausible. So if your maximum score is coming from here, that means a substitution is more plausible than a gap on J or I, and in these two cases, it will tell you the gap is more plausible than a substitution, right? So whatever is the highest score, we use that for computing an alignment score at I comma J, right? Now it can have different combinations, right? So as I said, we start off with this particular notion, right? So we need to have the alignment score. If we want to find the alignment score at I comma J, we need to find the alignment score before that, I comma I minus 1 comma J minus 1, right? Now if you start from this first position, right, say for example here, so you're trying to start from here, right? So the thing is, you need to initialize because you are moving from. So if by the algorithm, if you want to find the alignment score of here, which is Si comma Sj, let us assume this is the ith position and this is the jth position, Si comma Sj, we need the alignment score for S minus 1 comma J minus 1, right? Because that is how the algorithm will work. Now for in the if that is the case, for the first letter of the sequence, we need to do some initialization. We need to have an initialization score based on which the next of the alignments will be calculated. Now we can start with 0. We have started with an initial set of 0. In this case, we have all the zeros, and then if there is a match, we have a 1, I think.
Sir,
Yeah.
this is for Walter and Smith, right? Because it's 0.
Yeah, sorry.
So this alignment is the Walter one, right? Local alignment.
Yeah, we're talking about local alignment.
Because it's 0
Waterman,
on the top.
you're talking about the Waterman algorithm, yeah?
Yeah, 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 sir.
Smith Smith Waterman algorithm. Yeah, it's the same thing. It's the name of the algorithm, but yeah, I'm not. I'm just trying to get you an idea about how it works.
Okay, okay, sir.
Yeah, yeah. The idea would be, in this case, we are initializing with a score of 0, and if it's a match, it's 1. If it's a mismatch, is 0. But this is just for an example. But in another case, what can happen is you can start with uh, a gap penalty of 2, a match of 2, and a mismatch of 1, and in this case these will not be 0. So this will be 0. This starts off with 0, but if you move right, there is a gap, so you put minus 2. If you move right again, there is another gap, so you would add minus 2 again, and so on and so forth. So the initialization does not remain 0, and it becomes a multiple of 2, and like it becomes a little bit more constrained. Why? Say for example, in this case, since these are all 0, you can start matching it from anywhere. Say for example, this A, you can start matching it from here, because in this case also it will be 0, and the alignment score will be 1. So the first row and the first column will have similar values. But in this case, it is not shown here, you cannot start from somewhere like here, because you already have a minus 14 here, and this would not make it feasible to be aligned here, because you will have a very high negative score. So this kind of constrains the starting point, the initialization point of the alignment between any two sequences. So both of them are used depending on the thing that you're trying to do, but this is a more accepted method, more like frequently used method that you will see, right? Now a little bit about the algorithm itself. A little bit about the algorithm itself. The time complexity is, the time is proportional to the product of the length of both of the sequence that you're trying to align. The space required is also proportional to the product of the length of the two sequences. Uh, but you can do some algorithmic modifications to kind of make it more optimized and less space consuming. by not storing the entries of the dynamic programming table that are no longer needed for computation. You just keep the previous and the current row only. So yeah, so you just keep whatever has been aligned previously and what you're aligning now. You do not need to compute the whole all possible combinations of alignment. Like you do not need to fill out the whole matrix. You just need to fill the matrix where your alignment is happening and where you're scoring the alignments. So by that you can bring down the space required by quite a bit, right? Now uh, you have gotten the score. Now the idea is to get the alignment, right? So there will be a sequence of substitutions, small indels leading to this optimal score. And so basically here it is represented. If there is a red, there is a match. The match spelling is wrong, sorry for that. If there is a blue, there is a gap in the horizontal sequence, as I said. If there is a green, there is a gap in the vertical sequence. And so these three things represent that. So this is the match. This is, you have introduced a gap in the top sequence, and this is the one that you have introduced a gap in the bottom sequence. Now how do you recover the path? The idea is that you would, like, after, if you compute all of the possible alignment scores, you would fill out the matrix and you would start from here, right? So while yeah, so while scoring we started from here, but while getting the optimal alignment we start from here. We start from here and we have three options to move. Like, so if we start from this block, we have three neighboring blocks to move to, and we will choose which block to move to depending on the scores of these three blocks. So if this is, say for example, +1, this is -1, this is 0, we move to the +1 box. And now from here there are now a lot of other possible combinations that are possible, that these three, if for example this is the highest score, we move here, then we move here, and so on and so forth, following the highest score of the surrounding of the neighboring boxes, right? uh, and if at some position several choices lead to the same maximum value, the path, there can be multiple paths. That is what it is saying, that the alignment can be visualized in multiple ways with the same score. So multiple possible alignments is multiple alignments is possible there as well. Like, now the idea is that if you just ignore the initial and the final gaps, so what you're doing is, so if we started off here, right? So these are the set of initial gap score that we have like we have taken, and from here we build on the alignment, right? But if you imagine that you do not require all those initial gaps, you do not score those initial gaps, what you try to do is basically um, uh, set them to 0, as is done here in the left hand side plot. And uh, then what you are trying to do is basically, you are not penalizing for gaps that are flanking one of your sequences on the left or the right. So here this is basically a sort of a semi-global alignment where you are ignoring all the gaps to the left and the right initializations. So basically the initial set of gaps and the final set of gaps, you are just disregarding those to find the most best possible match there. So that is what is defined as semi-global comparison or semi-global alignment. So you set these things to 0. So all the 0 comma I, so 0 comma 1, 0 comma 2 till 0 comma n you put to 0, and here also you set them to 0, right? uh, I'll talk about the final gaps in a moment. Exactly. Right. Yeah, so here, as I was saying, that we ignore the flanking gaps by assigning 0 to the initial gap penalties. To ignore the final gap penalties, we choose the highest scoring entry in the last column or the last row and trace the path back from there. So we choose any of these. So in the earlier case, we were starting from, in this case, in this diagonal end box, right? But here, when we are ignoring the gaps, we can start from anywhere. We can start from anywhere in this last row, or we can start from anywhere in the last column. because there will be no gap penalties for the initial set of gaps, and for this case the final set of gaps as well. Now the idea is that the gaps, however number of gaps we introduce in the sequence alignment, but biologically they need to have some meaning, right? So you cannot just keep on introducing big gaps or like even smaller gaps inside the alignment, because biologically some things are more plausible than the other. So say for example, these two alignments, right? These two alignments look very similar. In the first case, there is a continuous gap of like 2. These two T's has been deleted. And in this case, the 3 uh, T's has been deleted, one leaving one after another. Now, if you just do if you just keep this algorithm, try to use this particular algorithm and have a constant gap score. So what will happen is it will tell you that the second alignment is the two of the alignment will have a will have a similar score, but you would not be able to distinguish between these two alignments because mathematically they're the same. Now, the idea is that we have an affine gap penalty. We have discussed this in the last class as well. So, the idea is to have an additional penalty for opening a gap. So, it would look something like this. So, this is your whole gap penalty. H would be your the cost of starting a gap, and K is the length of the gap, and G is the constant by which you are penalizing each base pair of the gap. We have discussed this earlier, but just to say that like if you want to open a gap, that should cost you more in terms of the alignment score compared to when you are just trying to extend the gap. So biologically, opening a gap is harder. Biologically, it is harder than to just extend a gap. So keeping that in mind, you have different affine gap penalties. Like now, if you just try to expand on the previous algorithm, uh, now we what we did, right? Just let me go back here. So yeah, what we did, we to compute the score for I comma j, we just took the best possible combinations of the three neighboring boxes here, right? So we take score all these combinations, and whichever is the maximum, we take that as the optimal alignment, right? But now imagine that you do not do it only for these three boxes, but you do it all for all the columns for j and for all the columns of I. So rather than checking for the best of the three values, we can check the whole of these rows, and we can check the whole of this particular column as well, and also consider different possible gap lengths, because you're starting from here, so there will be gaps if these two sequences are similar. So this particular alignment and this particular alignment will have a lot of gaps, so you need to compute depending on whatever is your gap penalty. So the idea is that we compute for all this set of values, for all this set of values, and then we find the max over the following max, right? So if so from this is the I prime here, and I think j prime is somewhere here, but anyway, for I prime comma j, minus the opening gap minus g into gap length, this is the affine gap penalty I was talking about, this equation, right? So basically, opening gap you have a penalty, and length of the gap you have a penalty for that again. So depending on the number of gaps here, you have a score for this. You have a score for I minus 1 j minus 1, and you also have a score for this. But try to see that what this is saying is basically maximum over I prime. So basically, you take all the three boxes here. In this case, it is 3. In some cases, it might be 30. You compute for all the 30 boxes there, and you take the maximum over that, right? And similar for j prime, you calculate all the boxes here, and then you. So I think I mixed up j prime and I prime, but anyway, like you need to compute for all the parallel rows and columns for this particular element, and then you take the highest for that. So this would be highest of all the blue rows, yeah, all the blue rows, and this would be highest for all the green columns, right? And then your original alignment, which will be coming from the diagonal, that will also be there, depending on whatever match mismatch is there or not. So the algorithm becomes a little bit complicated, where this is same, this is the exact same as what we had before, but in the next two cases we are taking the maximum over a set of values, not the a particular value itself. So in this case we had taken a particular value, just here to here what is the alignment score, here to here what is the alignment score. In this case we are doing all the set of possible combinations here, all the set of possible combinations here, and what is the maximum out of all of this, right? In both the cases. Okay, and
Sir, what is the advantage of this taking a maximum score than just looking at the top number?
so it can happen. So as I was saying that this particular, so the idea is that how just a second, let me find a good uh, yeah, I think this would be okay. Let's try it from here, right? So sorry, yeah, here on I'll try. So the idea is that in the previous case, in this particular case, you are assuming that the alignment can come from these three possible corners, right? Either there is a gap, one single gap here, or one single gap here, or there is a substitution here. These are the three possible combinations that you are thinking of, right? These are the three things that are possible by this particular algorithm. What this particular algorithm, the other algorithm, is doing is basically it is saying that it is it is possible that it comes from these three boxes, but it is also possible that there is a big gap here, or there is some other alignment that we are not considering. So these three boxes we are not considering in the last alignment algorithm, right? Also these three
Yes, sir.
boxes,
Yeah.
also these three boxes. So here what we are doing is we are opening up the possibilities that there can be a larger gap possible in either of the sequences, and we should consider that and also align and try to see what are the all the possible combinations and then take the maximum, not just limit our combinations to these three blocks. What we are doing is we are opening it
Okay,
up,
sir.
and we are trying to say that there can be big gaps possible, and if the score of the big gap is better than the substitution that we are doing, then maybe the big gap is the better alignment, right? So that is what is limiting in the earlier algorithm. Here we are just expanding that algorithm to include a lot more gaps and for the alignment.
Sir, nature, but nature doesn't prefer gaps,
Yeah, it
right?
does not, it does not, it is true, but it is possible that these set of the diagonal set of alignments that you are doing, these are mostly substitutions, and this substitution negative scoring for this substitution adding up is worse than having a gap, longer gap. Getting my
Okay,
point?
sir.
That is possible, right?
Yes, sir. Yes, sir. Got your point.
Yeah.
Yeah.
Okay. So yeah. So and the time and the space complexity becomes much more here. It is proportional to the cube of the sequence, so it is much more intensive algorithm to do. But Needleman, Wunsch, and Smith-Waterman algorithms kind of follow a more a similar approach to this than the earlier one that I was showing. Yeah, this is basically an extension of this. This kind of explains the algorithm. So yeah, this is your affine gap. These are your two sequences, two comparisons, and like these are the so basically this is the first condition, if you remember here. Yeah, this is the first condition. uh, this is the best possible alignment of Si and Sj. uh, there is another alignment which is, say for example, the gap here. So let us three possible cases. One is this particular alignment, one is this particular alignment, and one is this particular alignment, like three alignments. So in the first case, if there is a match or a mismatch, depending on that, that is the best possible alignment. The other is one the other one is the best possible alignment with one of the sequence that ends in a gap, and another one is the best possible alignment with the other sequence that ends up in a gap. So all the three combinations, so this all the three combinations we need to try. And what it in if you implement this kind of an algorithm, what you would need is three matrices like this to to basically compute the scores across these three different conditions. You cannot compute like for each so you cannot compute for each box and put it in the same matrix, because there will be no space and no connectivity in the matrix itself. So you make three different matrices, each matrix aligning to each of these conditions. So first is this, second is a gap in one sequence, and the third is another gap in another sequence. And then you compare the scores of these three, and whatever is the maximum, you choose that. The idea is the same, it's just that instead of using one matrix to store the values, we use three different matrices to make it more simpler and easily understandable. But it increases the space requirement by quite a lot. That is also there. Right. Okay. So this I'll not talk much, but yeah. So different type of initializations can be possible. So you can do an initialization at 0 0 to be 0, at I comma 0. So basically all these things. So here all are 0, but what it the that slide is proposing is basically we start with a 0 here, and all rest are infinities. So that is one possible thing that you can try as well. Sorry. Positive negative infinity for all I comma 0 and for all j comma 0. So infinity is assigned where there is no alignment possible. And then you can have different affine gap penalties. So this is basically the 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 linear function, because your basically gap penalty is scaling linearly with the length of the penalty itself. In some cases, what you can do is you can have a logarithmic scale for, say for example, you yeah. So I'll give you a small example. So if you have a same gap opening penalty for two sequences, two gaps, which is one which is half the length of the other, the score of the larger sequence will almost be double sorry, double negative the score of the smaller gap, right? So but biologically it does not make sense, because if you have opened a gap, if you are having, after opening a gap, if there is a gap of 10 base pairs, versus after opening a gap you have a base pair of 20 sorry, a gap of 20 base pairs, the thing is the second one is not double the difficult of the first one, because once you open the gap can be extended in a very easy manner in nature, because once there is a deletion, right, the ends of the breakpoints of those regions can be moved further away, can be, the deletion can be expanded, right? So the expansion of the deletion is not as difficult as to just initiation of the deletion. So what they do is basically they instead of having a linear increase, what they do is a logarithmic increase, where the in with increase in length of the gap, the increase in the penalty is considerably lower if you have a uh, linear kind of an equation like this. So that is there as well.
So if the length of the gap increases, we lower the value of the gap penalty.
No, we do not lower. We What we do is we instead of k we use a log k.
Okay, sir.
Yeah, or something like of that manner, right? So when your k increases, after a certain point it becomes like it like log 1,000 versus log, say for example 10,000, the difference between that and versus a log 10 versus a log 100, 10 versus 100 difference is much more compared to 1,000 versus 10,000, right? So what happens is, like, if you are increasing your gap penalty to a certain extent, the log will kind of undersell the penalty and it will reduce the penalty. So if you have something of 20 base pair length versus something of 40 base pair length, the 40 base pair length will not be exactly double of the 20 base pair length. It will be somewhat closer. It will not be double, it will be more, but it will be somewhat closer. So as and when you increase the length of the gap, if you use a linear scale, it will just linearly increase. But if you use a logarithmic scale or something like that, it it is basically diminishing returns. The more you increase, the less the difference becomes, right? So use something
Okay,
like a
sir.
g log k. Yeah. Okay. Yeah. So yeah, we have, so all the things that we have talked about are mostly global alignment algorithms, but can be used in local alignment as well. But yeah, mostly used in global alignment algorithms. Now, the idea in local alignment, as I was saying, that you are trying to match a small portion of the two sequences, not the two sequences completely. Now, if you do global alignment on these two sequences, you'll find a sparse alignment like this, a very few matches in between, a lot of gaps, and the match is not very good. But if you do a local alignment on this and you try to only match a small region of the two sequences, you find a much better alignment like this, right? So the if you do it on a dot plot, it looks something like this. So you have a global alignment which is there. It's not bad, but if you see a local alignment, it's very clear. It's a single straight line. uh, uh, yeah, so one good example of this would be a gene from a Hox family. So these are your homeotic genes. They have very short but highly conserved subsequence, which is called the Hox domain or the homeo domain. The If you do global alignment and try to find the best possible alignment, you can miss this particular local similarity that is there. Okay. Now we've talked about mostly pairwise alignment. We are talking about alignment algorithms or different techniques where we are just aligning two sequences. Now we talked about multiple sequence alignment where you are trying to align multiple, like three or more sequences all at once. For that case, if you are using a similar type of algorithm, excuse me, which is an exhaustive algorithm that you are computing for all possible combinations and they are choosing the best one, right? So if you want to do that in a multiple sequence alignment, you need a very big data structure, a multi-dimensional search matrix. Depending on the number of the dimensions will depend upon the number of sequence that you have. Uh, so if you have n number of sequences to align, you'll have an n-dimensional matrix, right? So this is good, but this does not, like, this is first of all computationally very expensive and it takes a lot of time and storage as well. So this is and kind of an impractical way if you are doing it on larger data sets, right? So dynamic programming algorithms are mostly limited for smaller data sets with uh, fewer sequence similarities between two sequences, right? Now you can use a heuristic approach for that. That is more what is used in algorithms of BLAST and other softwares. So this is a tree-based algorithm. So basically, it checks on multiple sequence alignment based on pairwise similarity, right? So first, what it'll do is perform pairwise alignment of all the sequences using some global alignment algorithm like Needleman-Wunsch, and it records the similarity score. Then it converts the score into evolutionary distances by creating a distance matrix, which is something like this. So all individual pairwise alignment, and then you do a sequence matrix for that. Evolutionary matrix for that. And then a guide tree is constructed from the distance matrix using a neighbor joining method to form something like this. So basically, which two sequences are more similar compared to the others. So A and B is more similar, so they are connected. Now AB is similar to CD, more similar to CD than E. So that is how this tree structure is basically built, right? Now after this is built, after when you have found that which two sequences are more similar than the others, what you do is then based on the positions, you identify closely related sequences and basically try to realign them. So basically you try to align them separately. So A and B separately you align here, C and D you separately align, and after these alignments they reduce to consensus sequences which are aligned to each other. So you now, since CD and AB are similar to each other, after the alignment of AB and CD, you try to align AB and CD. So AB is a aligned sequence, CD is another aligned sequence, and you try to align these two set of aligned sequences, right? And then you put in the E at the last because this is the with the lowest similarity to that, and then you do this method and to find the complete alignment. Now this method is good, and you have different algorithms and scoring matrices for that. Cluster is a very well known algorithm for multiple sequence alignment. It follows something of this algorithm of a heuristic approach as well. But I think the general idea is more important than the software itself. So the idea is that what you're trying to do is finding first individual pairwise alignments, which two sequences are the most similar. Based on that, you try to align those two sequences together, and you find all these pairs of sequences, and after you get this pair of aligned sequences, you try to align these pairs with each other, and you keep on iterating this depending on the number of sequences that you have. So this is a better way and a computationally more effective way to do this alignment compared to something like this, something like a using n-dimensional matrix with dynamic programming. Okay, now we come to the final portion probably. Is BLAST. Like I'm sure a lot of you are familiar with BLAST. A lot of you might have used BLAST, and Sir has also explained quite well in his lectures. So I'll go through it once in terms of what I think you should know in terms of BLAST. But if you have any so if you have any curiosity and want to know how it works, the best way to do is just go and experiment on the tool itself. It's freely available. It is very easy to use. Uh, the the options and everything are very easy to handle. So my suggestion would be, if you have not used Blast before, I suggest you go and give it a try. I don't think it'll be like that difficult for you, because it's a very easy to use software and a tool, right? And there are different types of Blast. So what is Blast? Blast is basically doing local alignment, right? The LA stands for local alignment. So Blast, first of all, is doing local alignment, and different types of sequences it can align. It can align nucleotide sequences, protein sequences. You can give it a nucleotide sequence and tell it, you can give it a protein sequence and ask it where in the DNA did this come from. So what it'll do is it'll have all the nucleotide sequences, convert them back into codons. From codons, they'll convert them back into what will be the DNA bases, and they find the DNA bases, and then they align it to all the genomes that they have in their database, right? So something like that is also possible, and there are others as well. So tBLAST10 will compare a protein query sequence to a nucleotide sequence. tBLASTX will compare a nucleotide query sequence to a nucleotide sequence in the database as well. So a lot of different combinations are possible, but mostly the BLAST10 and the BLASTP are the mostly used. So you have a nucleotide query sequence and you search in the database which organism has this nucleotide sequence come from, or you have a protein sequence and you want to find which is the protein this is most similar to in across different organisms. So yeah, mostly these are the use cases. I'll go through the steps one by one. So first, what it does, so when you give it a sequence, what it does is basically it'll create a lookup table or a list of words from the query sequence. This is called seeding, right? And the BLAST tool takes the query sequence, breaks it down into smaller sequence called words. For protein sequences, each word is 3 amino acid long, and for DNA sequences, each word is usually 11 nucleotides long. And there, if you notice, there are overlaps. So GTQ, TQI, QIT. So there are overlaps between these sequences. Same in case for the nucleotide sequences, and these are called k-mers. K is basically the number of elements in this sequence, and in this case this is 11. So it is an 11 mer, and you form these are all overlapping sequence. So you form a sequence from the first position, then from the second, then from the third, fourth, and so on and so forth, and you make a lookup table. So what is a lookup table? It's basically a data structure which will store that this is a sequence that is coming from offset 0, which is position 0 of the string. This the second sequence, this is coming from position 1 of the string. The third sequence is coming from position 3, and so on and so forth. So it'll create a whole dictionary, and basically a lookup table, not a dictionary, of all the sequences and where they are originating from, what is their offset, and what is the sequence that is there, right? So in the second step, it will do a database search of known sequences to find any sequence that contain the same words in the query sequence. This is done to identify database sequences containing your particular sequence. So you have, this is for proteins, this is for nucleotide. So in this case, there's one exact match here, and here there are two matches of your, so these are your, remember, this is not your whole sequence, right? This is your k-mer, and in this case these are your 3-letter um, uh, sequences that you have formed from here, right? So this and this, so you are not trying to match the whole sequence to whatever there is in your database. What you are doing is you breaking down into smaller sequences and trying to find where these smaller sequences are in your database, which is easier to do in terms of computation. Then it will score the similarity of these matching words depending on what you are what sequences you are comparing, and depending on that, it will use the different scoring matrices, PAM or BLOSSOM, or for proteins, for nucleotide sequences, it can use a scoring matrix of its own, and like with different combinations scored differently. So purine to purine will be scored differently, purine to pyrimidine will be scored differently, and so on and so forth. So this is how it is done, and in the final step, it will do the pairwise alignment by extending the words in both directions. So if you have a k-mer that starts from 1, then if there is a match there, so you take the k-mer that starts from 2 and try to match it there as well. If that matches, you take k-mer 3, k-mer 4, and so on and so forth, right? And basically the resulting aligned segment pair without the gaps is called an HSP, which is the high scoring segment pair. And an important thing here to note is that it also calculates a statistical significance value for each alignment, which is the expected value or the E value, right? So what does E value tell you? E value represents the probability of obtaining a sequence match by random chance. What it means is basically if you have a lower E value, that means the sequence match is less likely to be a result of random occurrence, right? So uh, if you have a lower p value, that means that there is a specific match of that sequence to whatever sequence it has matched to. So that is a good alignment. So you would want your alignment to have a lower e value uh, for higher level of significance. Here I have described a little bit about the e value as well, because it is quite interesting. Yeah. Uh, Sorry, excuse me. As I was saying, that it is an estimate of the expected number of random alignments with a particular score, or better, could be found by chance in a given database search. So what it is telling you is your sequence matching your database query, whatever be the query, database sequence. Does your sequence match the database sequence just randomly by chance, or that it is a very good match and there are not other matches anywhere in the database? That is what it is telling you. That is there by chance, if there is there a possibility that by chance this sequence can be aligned to 5 other different sequences as well. So that is what the e value will tell you. So the e-value describes the number of hits we expect to see by chance when blasting a database. It helps us understand if our hits are relatively unique or not. So is your hit found in only one organism, or there are other possible alignments with 10 other organisms that you are finding? So that is what the e-value will tell you. Or not organisms, or proteins, or different genes, or whatever you are trying to align. So an e-value of 1 means one expect by chance to see one match with a similar score. We need to be careful while interpreting e-values and all with respect to the biological question and so on and so forth. I'll not go into very details. So one important thing is there is no fixed threshold. It's a significance. So whatever, depending upon whatever you are willing to accept, you can set a cutoff, right? Uh, and also, a particularly a stronger e-value does not necessarily imply a stronger evolutionary relationship. It is just telling you that there are not many other sequences that this particular sequence is matching to. Only one, uh, the result that is coming, that is the best match possible, and the other one are not very significant. That is what it is telling you. It does not tell you anything about strong evolutionary relationship or functional or structural conservation or anything of that sort, right? For that, you need to do multiple sequence alignment and see the residues which are conserved and try to get an interpretation out of that. E-value is not for that, right? Now, how is it calculated? It's a simple formula where basically kmn e to the power negative lambda s. So k and lambda are basically your parameters from for these are called uh, Kerlin parameters. So these parameters are derived from larger data sets. You fit this model to a very large data set, and then you do some maximum likelihood estimation or something like this, and get these particular parameter values. Uh, S is basically the alignment score for that particular alignment. M is the length of the query sequence, and N is the length of the database. That is the sum of all the lengths of the sequence in the database. And this is how the E value is calculated. And more importantly, the E value will depend on the database size, because if your N will depend on the size of the database. So if you have larger databases, there is more chance of producing the alignment you see by chance. So the E values for the same amount of similarity end up being weaker, which is the higher value. So if you have a smaller database, your E value might be biased and be low. And if you have a very large database, the E value may again be biased and be very high, which is wanted. So you need to have an optimum length of your database as well, right? So yeah, so that is all in terms of the recap that I wanted to give. We'll move into the questions. Before that, if you have any questions,
Back to the
yeah,
previous slide, there was some kind of parameters,
yeah.
at artificial parameters. What are they? Can you re-explain?
So basically, uh, so when you make a model, right? So there will be parameters of the model. This is basically a model, right? So E equal to kmn e to the power negative lambda s. Now, there these parameters are constants which are specific to the database itself. Now, how do you calculate this constant? What you do is you fit this model to a large data set, possibly of your database only, and then you know the e value for the different sequences. You know you know m, you know n, you know s. M and n are the lengths, s is the alignment value, and so on and so forth. And then you calculate k and lambda from the data itself. So whenever we talk about parameter estimation, it is basically your model will have different parameters which are constants. These are not variables. These are not neither the independent variable nor the dependent variable. These are constants in your model. And how do you estimate those constants? You fit your model to a data, and depending on the data, you estimate what is the value of k and lambda, which perfectly fits this model to the data itself. So this is a whole
So
field of,
it's done
yeah,
experimentally.
it's not done experimentally. The data is experimental, but uh, you're just fitting this particular model to the data, and you see that what is the best value of k and lambda for which your model satisfies the data. Am I
Okay, sir.
am I understandable? So there's a whole uh, field of statistics that does this. So if you want to look up estimation
Okay, sir.
theory. The estimation theory deals with all this. So there are a lot of ways that you can estimate these parameters. There are Bayesian approaches where you need a prior understanding of how these parameters should look like, and then keep on changing little by little to fit the data best. Or there can be naive approaches like maximum likelihood estimation, where you just try to find the parameters by default with no prior assumptions of that. So there are a lot of ways that you can do it. It's statistically a little complicated. I wouldn't go into that much details, but if you are interested, please look up estimation theory.
Okay, sir.
Yeah. Okay. Yeah. Any more questions? Okay, then we'll move on to the older assignment questions. Okay. Just a second. I'll just. Yeah. I'm just turning off my camera for a bit. Yeah, I'll give a minute. Yeah. One more thing I wanted to confirm before I explain this question. Are you also attending the live session that is done by my colleague on Monday?
Sir, I am.
Okay. So I just have a quick question. Are the questions that I discuss exactly the same as what he discusses, or there are other differences?
Sir, there are differences. They're similar, but not exactly the same.
Okay, okay. I was just confused that if we have access to the same set of questions or not. So, okay, okay. Thank you. Yeah, coming to this question, what you want to do for sequence motifs? Motifs are basically conserved regions in the genome. Uh, those can be conserved for transcription factors to come and bind, some other proteins to bind, DNA polymerase to bind, and so on and so forth. So these are short conserved regions within larger sequences, mostly within genes, different genes in the genome. So local alignment is the better way to do because you want to find that small sequence similarity rather than the whole sequence being aligned to the bigger sequence. So yeah. Okay. Yeah, the answer is all. The GI ID, by the way, is Gene Info ID. This is an NCBI ID. FASTA sequence, I hope you know. It's basically it's it's a format in which a sequence of amino acids or sequence of DNA is written in, and UniProt is the database for the proteins, and ID of that is also accepted. So yeah. Okay, I see a couple of C's and a couple of B's. Okay, wait for another minute. Okay. Uh, okay. I think I. Oh, oh. Just give me a moment. I think I put the wrong screen as wrong. I did not put the answer here. Just give me a moment. Yeah. The answer. Sorry. Yeah. Yeah, the answer is C. Basically, 62 represents 62% identity, which avoids over representing of very similar sequences. So if you take all the sequences that are very similar in nature and then you try to create a scoring matrix out of that, uh, it is not going to go well because it is going to have a lot of bias. So they reduce the threshold to allow for at least 62% identical sequences uh, in terms of proteins, and then from there they try to build, like, if two sequences are already 62% identical, then what are the non-identical residues in these proteins, and from there they try to identify which are the substitutions that are more impactful versus no, less impactful, and give scores to them. So if you have 100% or like very high similarity, it is very hard to construct a matrix, scoring matrix like this. On the other hand, if you have very low similarity, very low identity in terms of two sequences, it is going to be harder, because if you have completely two different sequences, it is very hard to identify like which are the conserved regions, because there will be no conserved regions, and which region changes have brought about evolutionary divergence and so on and so forth. If you have no if the two sequences are very dissimilar, then you cannot find this set of matrix. But again, if it is too similar, it is highly biased. So you need to have some middle point. So 62 is the ideal one. In blast p as well, they use blossom 62. So yeah. Okay, I can see a lot of A's. Okay, the answer is A. uh, why is why is it the most acceptable one? Because these are both negatively charged amino acid. They are very similar chemically. So this makes a conservative substitution. So if it would have been positive to negative, it would not have been conservative. But in this case, both the amino acid are very similar in structure. In chemically similar in terms of the chemical components, and also similar side chains. So this is a conservative substitution. Yeah. Yeah, the answer is C. It depends on both the length of the sequence that you're trying to search and also number of sequences in the database. I hope you saw that from the E E value calculation as well. So yeah, yeah, that is true. So I think this is pretty self-explanatory that if uh, if it is a highly conserved region, then it is evolutionary. The evolution rate at that position is very low. It has not changed across different generations, different organisms, so it will have a high conservation score. So yeah, I hope you can see the full of the screen because of this. I can't seem to place it in a good place. I think now you'll be able to see. Yeah. Oh. Okay, I'll wait another minute for this. Okay, I see a few fours and few five. Okay. Um, I think the correct answer is four if I am not wrong. Yeah, four. But the people who have answered five, I think what you're doing is you're moving from this to this. So uh, that is okay. So I'll think I'll okay. So I don't know if I can put this side by side. Just let me try once. Yeah, I think yeah. Okay. So there are three options possible, right? So you move from here to here, or here to here, or here to here. Three possible options, right? So the diagonal one, you move from this to this. So this will be 0. Then there is a mismatch here. A and T, there is a mismatch. So there is a -1. So this will be -1 if you move from here. Now if you move from this position, this to here, there is a gap here because there's already an alignment here. So there is a gap here. So 0, and for the gap penalty is -2. And if you move from the left, from 6, then you move to the right, there is a gap here because yeah, you're aligning A to T, right? So there's a gap here. So I think the people who answered 5 did not consider that this is not a mismatch, this is a gap, right? Because you're moving you're moving from this position to this position, so there is a gap that you're keeping in the first position. So in the second in the third option, where you're moving, there is a match, and then there is a gap, then the t is matching, so that will be 6 plus -2, which will be 4. So 4 is the answer. Yeah, somebody was saying.
So,
Yeah.
yeah. So if 6 was in the place of 0, like in A position,
Hmm.
then would it be, should we consider as a mismatch then to take -1 and not gap of -2?
So you're
Like
saying
if it was diagonal traceback, then should we take it as -1?
if it's a diagonal trace
And if
back,
it,
it's always a match or a mismatch. If it's coming from the left or the right, it's always a gap.
it will be a gap.
Yes. So if you're moving from the
Okay.
diagonal, it's a match or a mismatch. If you are moving from left or the top, you're always putting a gap.
Okay,
Sir,
sir.
can you explain this place once more, sir? This part.
Okay. So there are three possible. Remember the algorithm that we talked about. So for getting the value of x, you can either move from the top, or move from the left, or move from the diagonal, right? That is clear.
Uh, yes, yes, sir.
Yes. Now when you are moving from the diagonally, when you are matching, it is always, there is either a match or a mismatch, because you're matching the same element there, right? So if there is a
Mm.
diagonal movement, there is always a match or a mismatch. When you are moving from left or moving from the top, you are introducing a gap. So what you are doing is, you are saying that this a matches with this t, right? So you are introducing a gap here, right? Or if you're saying, if you're moving from here, so if the, basically what you're saying is, here there is a gap, there is no match here, so there is you're getting a gap from when you're moving from the top, you're getting a gap where you're moving from the left. The
Yes,
only possible match or mismatch is coming from the diagonal elements. So
diagonal.
this is something that you can remember. So when you're moving diagonally, always look for matches or mismatches. If you're moving horizontally from top or from the left, it's always a gap.
Mm.
I'll just quickly go back to the slide that I discussed this. I think that will make it a little bit more clear. Yeah, remember this slide. So when you are moving, s minus 1 and j minus 1 will be the diagonally previous diagonal element. Agreed?
Hmm.
So if when you are doing that, you are adding a score of match or mismatch, right? But when you are moving
Yes.
from any of these left or the top elements, so this is basically I and j minus 1 will be the left element, right? You're introducing a gap
These are
here.
the horizontals.
These are all your horizon. The next two are your horizontals. When you're moving horizontally, you're always putting a gap. When you're moving diagonally, you're putting a match or a mismatch.
Mismatch or mismatch.
Yeah.
Yes, sir. Thank you, sir.
Okay. Uh, yeah, let's try this. Okay, I see only one answer. I'll give another minute. Okay, I think we'll discuss this. Okay, the idea is to have the coverage. By coverage, what it means is like how much portion of the second sequence covers the first sequence. What is the length of the uh, I ignore the this pause thing. I think this is like has not been rendered properly. The basically it has done it like 1 2 3 4 5 6 7 8 9 0, then again 1 2 3 4 5. But yeah, basically these are 10 11 12 13 14 15. This has not rendered properly, so ignore that. But the idea is that how much after the alignment, how much of the sequence 2 is covered in sequence 1. So basically if you do the alignment, it is all the alignment is mostly done. They have given you the alignment, and it basically ends at this particular position 12. So it calculates the basically 12 residues that are matching, not matching exactly, but 12 residues that are covered in the top sequence from the bottom sequence, and the total number of residues is 15 for the query sequence. So it's 12 by 15 times 100, which is 80%. I'm not sure where you got 73% from, but yeah, 80% is the correct answer. It's simple. It looks like a complicated thing, but it's just telling you how much of this sequence is covered in this particular sequence. So 12 residues out of 15. So yeah. I have not discussed this explicitly today, but Sir has discussed this, so I'll hope you'll be able to answer. Okay. Any other answers? Okay, I see a few D. Okay, let me discuss. The answer is not negative 0.21. I think you have only considered the first portion of the formula and not the second portion, so that is why you are getting negative 21. So the formula is summation of fi log of fi, where f is the frequency. So in column 4, basically you have 3s and 1a. So there is 75% for s and 25% for a, right? So the fs becomes 0.75 and fa becomes 0.25, and then you do summation of this. So 0.75 log 0.75 plus 0.25 log 0.25. So I think you have missed this portion, the people who are answering negative 0.21, but you also have to consider the other one for the frequency as well. So if you add this up, this comes to negative 0.56.
Sir, in our exam, do we have given a scientific calculator to calculate the log scores?
I'm not sure, but I'm not sure
So do
about.
we have to remember it
No, no, no.
or a
I don't
table?
think you need to remember. If you need log values, I think they'll provide. Like if some question
Okay,
needs any
sir.
log value,
As in this case, it was not provided,
it
right?
was not provided here. But I think in the exam,
Uh,
if you require a log score, they'll provide you. So yeah.
Okay, sir. Because I got confused because of the log score, so that's why my answer was a little bit
Okay.
wrong.
Okay, no problem. But I think if either in the exam, if you do not have a calculator, either they'll give you option of an expression like this, or they'll give you the log scores. So it doesn't. Don't worry about
Okay,
that
sir.
much. Yeah. Okay. Okay. Last question. The answer is B. You expect if you have a lower E value, it tells you that is a significant uh, alignment and it is not by chance. So yeah, it's false. Okay, I think that is all from my side. If you have any questions, do ask.
Sir, I had a little bit problem about how to construct a PAM matrix in the first lecture of Michael Krumha, sir.
Okay.
So it was confusing. The last part was a little bit confusing.
Uh, okay. Let me bring it up. Uh, any other questions also? Like I'll bring them up together then.
Sir, I am not good with log scores because I was not with a math background. I skipped 11, 12 math, and I'm just in BSc second year, so l- I have a problem with how to calculate the log score. So how do I
You
learn the
don't
log
don't
to
have to worry
calculate it?
about that because log is just a scale, right? It's just it's a function which scales your value. For every value, there is a score for the logarithmic score of that particular value. I don't think you need to learn what logarithms are. It's just a scale. And for so basically if you have larger values you and very sparse values, so if you have say values for example 1 to 10 and then also you have 10,000, 20,000, 50,000, it is the scale of the value is too large, so to bring down the scale you use logarithmic conversions and so on and so forth. But I don't think you need to worry about logarithm. They'll give you the values if required, and I don't think like it's not a big deal. It's just a scale.
Okay, sir.
Yeah.
Okay, sir. So do I don't have to go in deep for this?
No, but just know what logarithm is. It's not a very hard thing to.
Yeah, no, sir, no, sir. I have a basic idea because in according to NEP, I have taken MDC maths, in which we have done statistics, and
Okay.
we also have done some integration, differentiation,
Okay.
also.
Okay.
So I have a pretty good idea, but I'm not good with log scores.
Yeah, it's just a scale. I don't think you need to worry about that. Yeah. For the PAM matrix, I think you're talking about this.
Yes, sir. Yeah, the last part, the Rij value.
Which RIJ value? Acha, are you talking with respect to SIR's PPTs?
Yes, sir. Yes, sir.
Oh, I currently don't have that. Uh,
Okay,
I'll
sir.
need some time to bring that up. So probably next week you can remind me.
Okay, sir.
I'll explain that. I don't have that right
Okay,
now.
sir. Okay, sir.
Okay.
No problem.
Yeah, yeah. Please
Sir, do we
remind
have
me.
to construct a PAM matrix in our
No, 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 no,
exam?
no.
No.
Uh, uh, since you guys are interested, this is a very good paper that I think you can read in terms of sequence alignment and uh, different. I think. Acha, acha, I think I need to reload this. Just give me a moment. Yeah. Yeah, this paper discusses a lot of different alignments, different types of alignments, local, global, what do you do in case of non-coding and coding regions, and so on and so forth, different types of blasts, global alignment, and uh, the maths behind it, and dot plots, and mostly it covers almost everything that we have discussed. Yeah, multiple alignments, scoring functions, yeah, different algorithms and these kind of things. So you probably can read this paper. It's going, like, the language is going to be a little bit difficult, but I think you'll be able to understand what different types of alignments do. There is another paper, PAM. I forgot the PAM matrix paper. Day of um, day of publication. There's basically the original publication I'm trying to find. I think these are not the original ones. Let me search in Google Scholar. This looks like it probably. I think this one explains the PEM matrix, but I don't think this is the original paper for that. Yeah, this is the one. Yeah, got it. Oh. The paper seems to be hard to be found, but okay. I think maybe, yeah. No, not this one as well. Okay, I can't seem to find the paper right now, but I think you can read this paper. This has a lot of reference to the original ones. If you can see if you can find it, but this paper also explains a lot of that. So yeah, if you're interested, please go through these papers. For Blossom also they'll have so they describe all the mathematics behind it, how they calculate, and what are the possible amino acid replacements, what are their values, and all. So these are like quite extensive papers. Don't, like, if you are not from a mathematical background, it's okay. You do not need to worry too much about it, about the mathematics, but try to understand what is happening. Yeah, so I think you are able to download the paper, right? Because this is, I think, behind a paywall. If you do not download
Yes, sir. Yes, sir. We
okay.
have. I have able to now.
Okay then, any more questions? Okay, that's all from my side then. If there are no questions, we'll close this session.
Sir, thank you so much, sir,
Thank
for
you,
this
sir.
session.
Okay. Thank you guys for joining. See you next week. And okay, I'll remember. I'll write it down. Your PAM construction, I'll talk about it in the next class.
Okay, sir.
Thank you.
Thank
Thank you for joining.
Thank you, sir. Thank you so much.